Few slides before we get started today. So yeah, this is the lecture on the visual system, week 5. For this unit, there will be no technical project or technical questions, but I think you guys still will have an assignment. So there will be that one synthesis question due Tuesday, and then all of the synthesis questions, all the other ones are due Thursday, if I'm correct. But yeah, okay. Here's the attendance form for today, so take a few minutes to fill it out. Okay, maybe give me like a thumbs up when you guys are ready. Has anyone not scanned the QR code? No? Okay, we can. Okay. Week 5 rankings. Okay. I don't know where the slide went, but I guess from last time you guys were asked what's the right way to put toilet paper. The whopping majority said over, which
like 25 to 2, or 29 to 2 or something. There was a clear consensus, and I also agree with that consensus. That is the right answer.
Yeah. So I guess that's the right way to anyone who picked the other way. But yeah, here's the TA rankings. I guess Sunflower is winning, Lavender, Daisy. I'll try to ask some questions, so you guys answer them. And if you have any questions, please ask me as well. I mean, just a disclaimer, I am on the Neuro side more than like the CS ML side, so I can definitely answer those questions better. But yeah, if everyone's good with the attendance, we can get started. Oh, hi. Okay, I'll just put this back up. Yeah.
The form is also in the resources page on Discord, so you guys can just click through there. If you ever miss it, you can just find it on the Discord.
Yeah. Okay, perfect. Okay. Let's get started then. So yeah, week 5, today we're going to be talking about the visual system. So I'm sure you guys probably had like a neuro lecture before this, but just a quick overview of the lobes of the brain. So we have the frontal lobe, which is responsible for movement, planning, and decision making. This is where you have your prefrontal cortex, your motor cortex, and also a part of the brain called Broca's area that's mainly responsible for language processing and kind of like speech development. Then you have your parietal lobe, which is kind of right behind it, and that processes sensory information, including touch, temperature, and pain. And that also manages something called proprioception, which is kind of your perception of you in the world and your surroundings. And then in the very bottom of your brain, you have something called the temporal lobe. This handles your hearing and memory. It includes something called Wernicke's area, which is for language comprehension. And then the left temporal lobe, it's also for language learning and memory. So also kind of responsible for language. And then lastly, on the back, this is the lobe that we're going to cover today. It's called the occipital lobe. It's dedicated to vision, and then it processes visual information, and it's involved with the visual cortex that we'll be looking at today. And then also just a quick overview of the neuron. Shapes and size may vary, but generally the structure is the same amongst all neuron types. So you have your dendrites, which are kind of these processes that take in information from other neurons. And then you have your cell body, which kind of integrates those signals and then decides whether or not to generate an action potential. You have your axon, which is where your action potential will travel down. And then you have your synaptic ends, which then will release neurotransmitters onto other neurons to either inhibit or excite pathways. Okay. And the visual system. Okay, so the official definition of the visual system, most people say it's a complex network that processes visual information involving the eyes, neural pathways, and brain regions dedicated to interpreting visual stimuli. So a fun fact, 40% of the human CNS is purely dedicated to processing visual information. That's how important it is. And I'm sure that kind of makes sense because a lot of the things you interpret around you are through your visual system. So yeah, there's mainly three parts. One, you have your eye anatomy, which we'll be talking about, which is kind of like your photoreceptors, rods, cones in your eyes. Then you have your neural pathways. So that's how information gets from your eyes all the way to your brain. And then you have visual processing in your brain, which includes parts like the optic nerve, the lateral geniculate nucleus, which we'll look later on, and then the primary visual cortex, which is in which lobe? Does anyone remember?
There was a good question on this last week, and I'm sure there was also one in the neuro lecture.
So which lobe is responsible for visual processing? Oh, occipital, yeah. Daisy. Okay. Yeah. There'll be more questions later on, so. Okay. I just thought this was quite interesting, but the Nobel Prize in Physiology and Medicine in 1981, it was divided amongst these three men. So one half was awarded to Roger W. Sperry for his discoveries in the functional specialization of the cerebral hemisphere. So, for example, your left versus right brain. And then the other half was given to David Hubel and then Torsten Wiesel. And these two were involved in information processing in the visual system. So that's kind of how big this discovery was in this whole system. So yeah, pretty important. Okay, this is a little bit more physicsy, but I'm sure many of you have probably seen this diagram before. It's the electromagnetic spectrum, and it's waves of energy traveling at the speed of light. And of course, they vary in how much energy they can carry. For humans, we mainly are able to see 700 to 400 nanometers, that wavelength of light, and that's what contains kind of the visible light or the rainbow that we're able to see. In terms of wavelength, violet is more shorter, so that's like more energy, and then red is your longer wavelength and also less energy. So, yeah. Oh my gosh, that was loud. Okay. So phototransduction, this also involves the electromagnetic spectrum, but this is the process by which how we're able to interpret light and then convert it into electrochemical signals that our brain can process. So of course, the light that we see is the electromagnetic spectrum within our range. And then for us, the wavelength of the light is our perception of color. And then the amplitude of the light is the perception of brightness, so how bright or dark a visual field is. And then, of course, the range that we can see is 380 to 750. So I think the previous slide I said 400 to 700, so it's around within that range. But yeah, so the main process, I guess, is you'll see light stimuli, and then the rod, cone cells, neural pathways that will convert it into electrical chemical signals, which are mainly neurotransmitters, and then your brain will interpret it in that way. Okay. Any questions at this point? Okay. So now we're going to move on to the anatomy of the eye. So I'll just go through and quickly run through the parts of the eye, the main parts. So first you have something called the cornea, which is kind of that transparent layer at the front of your eye, and that's what lets light onto the eye. And the cornea is kind of like this flexible lens, so that will actually refract the light rays to make sure that the direct light hits the retina, which is near the back of the eye. And then you have your lens, which is also responsible for helping direct light on the retina. It has less refractive power than the cornea, but it's also advantageous as it's connected to ciliary muscles in your eye, so its shape can be modified. And that can help you focus on things that are either close up or farther away. And then as people get older, your lenses, they naturally become less flexible. So that's where you develop kind of like the nearsighted, farsightedness for which you need glasses, or like reading glasses specifically. And then you have something called the sclera. So that's like the white, tough outer covering that protects your eye. That's the like white part you can see. And that's kind of a bit more firm, and that helps maintain the eye's round shape. And then you have the iris, which is your colored part of the eye, and that helps control the size of the pupil. And the pupil is that little black dot within the center of your iris, and the pupil, its size can be adjusted to kind of modulate the amount of light being let into your eye. So within like really bright rooms, your pupil will kind of contract to let in less light, and then if you're in a darker room, it will dilate to try to kind of like maximize the amount of light that's entering your retina. Um, and then you have the ciliary body, which is a ring of muscle uh, that helps like adjust the lens curvature. And then yeah, you have your retina, which is the part of your eye that actually contains neurons. And it is technically part of your eye, but it can also be considered part of the central nervous system sometimes. And its main function is to detect light and then use that light to produce those electrical signals that eventually go back to your brain. So within the retina, we have this part called the fovea, and that's where so we'll later in the lecture learn about two types of photoreceptor cells, rods and cones. And in the fovea, there's many more cones than rods. And then at the center, there's this part called the foveola, which there's no rods at all. So because of the high cone content in the fovea is a part of the retina that has the capacity for the highest like acuity of vision. And that's actually what like causes you to move your eye so that like you can focus your eye on a certain field of vision to get in the most amount of information possible. And it's also like important for discerning certain details about your visual field. And then lastly, kind of near the back of the eye, you have the optic nerve, which is what transmits visual information from your retina to your brain's visual cortex. Then you have your optic disc, which is an area where the nerve cells actually leave the eye. So there's no photoreceptors here. So that's what creates something that you might have heard of called the blind spot in your eye. And then last.
Okay, that's an alarm for when it's time to switch classes.
Class isn't over yet, guys. But so then you have something called your vitreous humor, which is this gel-like substance that fills the main cavity of your eyeball. That kind of just puts pressure on your eye, also helps maintain its shape. Then you have something called the aqueous humor, which is this fluid between the cornea and the lens, and that also helps maintain pressure and kind of keeps your eye moist. And then the conjunctiva, that's a clear thin membrane covering the inside of the eyelids and the sclera, and that keeps your eye moist as well and protects from infection. So yeah, I know that was a lot and really quick, but for you guys to look through later, I also put the definitions here as well in the next two slides. So feel free to look at that. Okay, quick question. What's the part of the eye that's responsible for like creating like a blind spot? I don't know who's saying one now. I think I saw hers first, actually. Yes, it is the optic disc. Okay. Okay. This is just a quick little. Uh-oh.
I think you want to be animated?
Oh yeah. Okay. Yeah. This is just a quick little video that I found on the internet, but like I mentioned, there's the blind spot. So, uh-oh. No, it is a video. Is it not? Oh shoot. Okay. Well, you guys can look back and maybe like try clicking on the video on your own time and testing it out, but you can try to just like cover one eye and then test out your blind spot. Yeah, that's okay. Okay, so does anyone have any questions on like the anatomy of the eye before we move on to neural pathways? Okay, let's keep going. Okay, so the retina. So your retina, like I said before, is a part of. Oh, my bad. Sorry. Okay. So the retina is the part of your eye that like actually interprets the light and then is able to send those electrochemical signals over to your brain. This is not on the slide, but just so you guys get a perception of how like light enters. So the retina is at the very back of your eye. So light will go through your eye, hit the very back of the retina. So that pigmented cell layer is the back of the retina, and then the information will actually process from back to front. And then it eventually goes to your ganglion cells and kind of leaves the eye. So light goes from front to back, and then information goes from back to front. So just keep that in mind. Yeah. I don't think you guys need to know that now, but that's just an interesting fact. So there's multiple layers in the retina. So the first layer that the light actually hits is what's called the pigmented cell epithelium. So that helps maintain the photoreceptor cells and then it keep them functioning properly. But another main reason you have that, actually, if you look at like a scan of your eye, that part of your eye is dark. And when light hits the back of your eye, it might like refract. And especially if you're in like really bright areas, that layer helps absorb extra light photons. So it gives you like a clear image basically. And then you have your photoreceptors, which are your rod and your cone cells. So this is also at the very back of your retina, in front of your pigmented cell layer. And it's responsible for converting light into electrochemical signals. So each contain like hundreds of these little stacked discs within them, and that's what's capable of absorbing the photons of light. And absorbing the photons of light, it causes the level of neurotransmitters to change and release in order to convey information about a visual scene. Um, so then you have something called bipolar cells, and that responds to the changes in the amount of neurotransmitters that are released by your rods and your cones. And that passes on further signals about perceived light. And then you have something called horizontal cells. So these have dendrites that spread horizontally amongst the retina, and they make contact with multiple photoreceptor cells. And this kind of modulates the function of the photoreceptor cells to do things like enhance contrast, for example, if you're in like a really bright room, or adapt to changes in lighting conditions. So then you have something called amacrine cells, and this kind of does the same thing. This makes contact with bipolar cells, ganglion cells, and other amacrine cells, and this is also involved with refining the visual signal through their ability to modify other retinal cells. And then lastly, you have your ganglion cells, which are kind of like the true neurons of your eyes in a way, and they receive the signals about the perceived light from bipolar cells, and then they carry the visual information through the brain. So the ganglion cells, all their axons kind of form together to form the optic nerve, and then those leave through the optic disc to eventually get to the brain. So yeah, any questions about that? Okay. So next, just a quick overview of rods versus cones. So these are the two photoreceptors we talked about in the previous slide. So rods, they don't provide for color perception. However, they're very sensitive to light, and then they have low spatial resolution, So they're not very good at seeing details. These are mainly the photoreceptors that are responsible for detecting changes in light. So in low light conditions, rods are the only photoreceptors that are actually activated. So that's why it's kind of hard for you to maybe like decipher color or details. And then when the levels of illumination or light increases, the rods eventually stop responding and then fail to convey information. And then the visual processing kind of gets transferred over to your cone cells. And then the high sensitivity to light causes them to be overstimulated or saturated even in normal room light or sunlight. And then, okay. Yeah, so in the conditions where you have like really bright light, the cones are the dominant photoreceptors. Surprisingly though, there are way more rod cells in your eye than cone cells. There's about 90 million rod cells. So I think some of you guys might have experienced this, but for example, if you go from like a really dark room to a really light room really fast, there's something called the black hole effect. And then if you go from a light room to a dark room, there's something called the glare effect that happens. So this occurs because the rods and the cones actually have different reaction times to changes in light. So yeah, and then that leads to like kind of that temporary inability to see in the dark. And then eventually your eyes get adjusted to that. And then the glare effect is caused by the rapid change in light intensity, and then that can kind of temporarily blind your eyesight. And then lastly, cones, these are the ones that enable you to see color, but they're not very sensitive to light, but they have like really high resolution, spatial resolution. So that's what causes you to see like the really fine details in your visual field. And there's significantly a lower amount of cone cells than rod cells. There's only about 4.5 million cone cells, and cones work very well in high light conditions where you can see color. And you have three types of cone cells. You have red cone cells, green cone cells, and blue cone cells. There's kind of that image over there. And those are what get activated in kind of different intensities to perceive color around you. Okay. Any questions about that? So if you're in a dark room, what type of photoreceptor would be activated? I saw his hand go. Rods. Nice. Sunflower. Dang, sunflower is going. Okay. I'll keep my eyes in the front row from now on. Okay. Lastly, I mentioned, okay, quick question. Are your ganglion cells near the back or the front of your retina? The front, yes, good job. Okay. And then light, does it go from the back to the front or the front to the back of your retina? Anyone else? Oh, right there. Front to the back. And then information? Yeah, back to the front. Okay. That was kind of just a general thing, I think. Uh, yeah. Okay, ganglion cells. You do have two types of ganglion cells. So you have something called M ganglion cells. These are larger, faster, and then these mainly respond to kind of movement in a visual field and then also differences in brightness. And these ganglion cells mainly get rod input. And then you have P ganglion cells, which are smaller, slower, and then these are the ones that are responsible for kind of detecting specific attributes of a stimulus, for example, color, shape, texture, and then these are mainly used during cone input. Okay. Are there any questions about the cells in the retina? Okay. So if you're in a really bright room, like for example, a sunny park, which type of ganglion cells are going to get activated? Okay. P ganglion cells. Nice. So far. Okay, visual systems in the brain. This is also going to be kind of a dense slide. Okay, I guess that's class starting. So this is also a dense slide, but I put more definitions in there for later. So you have your two eyes. This is actually a kind of a funny thing about, so humans have binocular vision. We have two eyes, and both eyes actually process information from both halves of your visual field. So for example, your left side, it will process information from the left visual field and also your right visual field, and then right side will do right visual field and left visual field. The funny thing is though, this information eventually crosses over in your brain. So your left visual field will get interpreted by your right side of your brain. Right visual field will get interpreted by the left side of your brain. So yeah. So in the previous slide, we talked about kind of how the ganglion cells eventually bunch together to form the optic nerve, and that leaves the optic nerve is actually one of our cranial nerves. If anyone, just fun fact. But the optic nerve extends back into the brain. It's below the hypothalamus in this part of the brain called the optic chiasm. That's kind of like the X shape you're seeing. And then at the optic chiasm, like I said before, about 60% of the axons actually cross over to the other side of the brain. And then some of them do stay on the same side. And that kind of helps us with our binocular vision, so getting a range of a visual field from both eyes. And then these fibers that are coming from the nasal part of the retina, they cross over. So fibers from here, whereas the temporal ones are the ones that do not cross over. Yeah, so then, like I said before, the result is opposite sides of your brain, opposite different parts of the visual field. So then after the optic chiasm, the fibers develop into something called the optic tract, and that extends into multiple parts into your brain. So some fibers go into an area in the brainstem called the protectum, and that's involved in a number of visual functions like the pupillary light reflex. So if you're in like a light room, your pupil contracts, and then if you're in the dark, your pupil dilates. Yes. So a lot of these type of like involuntary movements, your brainstem is responsible for. And then the other fibers go to a region of the hypothalamus that's called the suprachiasmatic nucleus. This is actually responsible for maintaining your circadian rhythm through interpreting like the amount of light. So that's kind of responsible for like saying, oh, it's daytime, I'm like awake, or oh, it's nighttime, it's like time to sleep. And then other fibers go to the part of this brain called the superior colliculus, and that's what helps the brain coordinate head and eye movements. So if you want to like focus on something, that's what helps you kind of turn your head, turn your body, and then maintain your attention on something that's going on. So like if you're watching a basketball game, for example, that's what helps you like follow the ball or follow the certain player. Okay. So that's like kind of minor parts that information from the optic tract goes to, but the main majority of fibers from the optic tract go to this nucleus in the thalamus called the lateral geniculate nucleus. It's like at the very bottom of that image. And then neurons from this region extend towards the back of the brain as bundles of fibers called optic radiations. So you have the optic nerve, you have the optic chiasm, you have the optic tract, and then lastly you have optic radiations that go to the very back of the brain. So these optic radiations, they travel back to the part of the cortex, which is in which lobe? Can anyone guess? Yes? Yes, it is the occipital. So the very back of your brain, they eventually hit the occipital lobe, and this part of this region called the calcarine sulcus. Um, Not really important to know, but just remember that this is part of the primary visual cortex. So eventually, all the information from your eyes will go back to a part of the brain called the primary visual cortex. And that's eventually what integrates all the information that comes from your retina, rod, cone cells, and helps actually form an image. Um, so there are many different types of neurons in the primary visual cortex, and these are activated and help decipher different parts of your visual field, such as orientation, movement, contrast, kind of like depth that you're seeing within the field. And then the PVC also communicates to a bunch of other visual areas around it. And then the neurons in this area, we don't know that much about them, but we think that they're specialized to some degree for also differentiating different aspects of a visual field. Okay. So these next two slides kind of just talk about everything I just said about the pathway. And then here is kind of different pathways within the primary visual cortex. So you have your primary visual cortex, and then like I said before, there are different groups of neurons that may decipher different parts of your visual field. So you have these sections V1 through V5. So V1 is your primary visual cortex. V2 is something called the relay station. So that's where like the integration of information starts, where your brain kind of starts combining different features, for example, color, shape, motion. And then V3, it's responsible for processing dynamic form, so shapes of moving objects. V4, it specializes in color perception and object form. And then V5 slash the part of your brain called MT, it detects motion, direction, and speed. So my figure kind of makes it seem like it's like a pathway, it goes from one to the other, but that's not really that true. All of these parts of your brains are like, all these parts of your brain are working together at the same time to interpret your visual field. So it's not like one goes from the other. So sorry about that. But yeah, okay. That was a lot. Any questions? Okay. So then lastly, two more pathways. So you have a dorsal and a ventral pathway within your brain, and this is kind of what goes from your primary visual cortex back to the rest of your brain and kind of what helps like move your body in response to your visual field. So you have something called a dorsal pathway, which is the where pathway. So dorsal, in terms of like body orientation, that means the top or the back of your brain. So your where pathway, it's from the back of your brain and it goes from V1 to V2 to V5 and then V6. Not too important, but so what I said before about like light coming in and then your brain going from like V1 to V2, that doesn't really happen, but when it's going back to the rest of your body, it does follow a pathway. So then this goes eventually to the parietal lobe, which is responsible for like your perception in your location, I guess. And then this helps with localization of objects in space. And then you have your ventral, which is your what pathway, and ventral means kind of underneath towards the bottom. And then this goes from V1 to V2 to V4, and then part of your temporal lobe called the inferior temporal cortex. And this is responsible for object identification and recognition. Okay. Okay. So if someone's like running towards me and I wanted to like be able to decipher how far they are from me, which ventral like pathway or which pathway do you think it would be? Yes. Dorsal. Nice. And then say there's like a ball and then I want to figure out like what type of ball it is. Ventral. Nice. Okay. Lastly, does anyone have any questions about the two pathways? No? Okay. Oh, yes. That's actually a really good question. I'm totally not sure because like your visual system's known to be one of the fastest systems in your body, so I'm not sure why it developed that way. But hmm, yeah, I'm not too sure. But maybe we just evolved that way. Yeah, it might have been like a protection thing. I don't know, but
It just like wasn't that big of a reason to have it.
yeah. Okay. Any other questions? That was a good question, though. Maybe someone will figure it out. But any other questions? Okay, we're almost done, I promise. Just a couple more slides. Okay, this is something that I just thought was interesting to talk about. We've been talking about the visual system a lot, but what happens in people who may not have that great of a visual system? So perception and blindness, that's a big part of research that's going on in people who study the visual system. And a lot of people may have complete blindness or like be described to have complete blindness, but in a way it's not total blindness. They are still very much able to perceive their environment, perceive different aspects of their environment, and they do so in multiple different ways. So light perception, some blind individuals, they are still able to retain a degree of light perception. They may not be able to see different colors or really like minute details, but they can still perceive like the amount or intensity of light. So that also helps them navigate a room and their surroundings. And then color perception, while total blindness does mean no color perception, individuals who may become blind later on in their life, they can remember colors from past experiences or they can recall colors when described by other people. And then touch and sound, I'm still not sure how this works, but there's this like common saying that if one person maybe loses one senses, all their other senses like make up for it. A lot of blind people, their touch and sound is very, very like elevated. Like they have really good senses in terms of touch and sound, and they can also rely on this, of course, to perceive their environment. And then echolocation, this is commonly used by a lot of animals as well, such as dolphins. And some people, they learn to use this as well to perceive their environment, such as making sounds or something like that. Like, for example, if a lot of people may have like a stick that they use to kind of interpret their environment. And then dreams, this is another thing that a lot of people ask, do blind people still have dreams? And their dreams actually do involve similar like visual fields as normal people as well. But the details of their dreams may differ based on their level of vision and then of course when they develop blindness as well. Um, so yeah, these compensatory senses, they enable blind visuals to interact with their environment and understand the world in ways normal sighted people, of course, cannot. Um, yes. Okay. Any questions on that? I do not know much about that field, but I think there's a lot of research going on, a lot of cool papers that you can find. Um, and of course, for people who are studying like regenerating vision or preserving vision, your visual system is very importantly plays a role in that. Okay, just this last slide, sorry with how the title and stuff came out, but it's kind of just a review of the visual system and then also comparing it to computer vision, which a lot of you may be familiar with. So computer vision, it's a field of artificial intelligence that enables machines to interpret and analyze visual data such as images and videos the way humans may do. So whereas we have, for example, the eyes, different pathways in the brain, computer vision, they use algorithms, cameras, sensors, and CNNs. And then processing style, we have parallel processing and holistic pathways. And then, of course, our visual field, like we have previously lived experiences, emotions, bias to different things, which help us perceive our visual field. But then computer vision, it uses sequential and mathematical pathways such as pixel values, feature extraction, and learned representations from maybe different images that you might feed a model. The learning mechanism, of course, it's very data driven, minimizes error on labeled data sets, whereas we kind of develop and learn throughout our lives to interpret our visual field, and then that's something that adapts continuously from birth. And then the limitations, of course, we're prone to optical illusions. We're affected by fatigue, attention. Some people pay attention more to other things, which also affects how your visual system processes. And then bias. And then computer vision, it's sensitive to noise, lighting, and context. And then it requires retraining to adapt. And then lastly, the goal for us, we have evolved to use our visual system for survival and interaction and navigating our environment. For example, recognizing faces, interpreting visual cues. And then for computer vision, you will see it a lot in kind of like self-driving cars, for example, to detect other cars on the road or pedestrians. A lot of people are using it to recognize handwriting, and in the medical field, especially, for example, interpreting different scans or diagnosing tissues for certain diseases. That's also becoming a very hot field. So, yeah, that's it for today. If anyone has any questions, I will try to do my best to answer them, and then we can move on to a Kahoot.
You explained everything so fantastic.
Oh, thank you. Okay. It was a lot. Of course, this is very much just surface level. There's so much more things to discover and learn about in the visual field or visual system. So yeah, if you guys like, I know Psych 202. If anyone's interested in taking that class, there's a wide unit that covers the visual system, so that might be interesting to take. But yeah, okay. If there's no questions, we can move on to the Kahoot. Okay. Thank you guys for listening.